Welcome back. This video session is on mutation and genetic disorders. The learning goals are 1. Identify 3 types of genetic disorders. 2. Describe gene mutation caused by substitution, deletion and insertion. 3. Differentiate between point mutation and frameshift mutation. 4. Provide 2 examples each for autosomal and sex-linked Mendelian diseases in humans. 5. Describe the causes of chromosomal aberrations in humans. And 6. Provide 3 examples of Aneuploidy in humans. As we have studied in this chapter, each and every trait in an organism is controlled by one or more genes. And we also know that genes occur in pairs, and these are located on specific sites of chromosomes. Now, an individual can be having a gene in homozygous or heterozygous combination. And in each chromosome, we can be expecting hundreds to thousands of genes. For example, the largest of the human chromosome, which is chromosome number 1, has over 3,000 genes, whereas the male chromosome Y has over 200 genes. Genes thus become the fundamental units of heredity, and an individual inherits one copy of each gene from the father and from the mother. Well, what exactly are these chromosomes on which genes are located? Chromosomes are organized bundles of DNA packaged in the nucleus of eukaryotic cell. So we know DNA stands for deoxyribonucleic acid. And we also know that DNA are large biomolecules. Suppose if we have to take an example of human DNA, if we have to completely stretch out our DNA, it has a length of over 2 meters. And such a lengthy DNA needs to be accommodating inside a tiny microscopic nucleus of our cell. So DNA is able to fit inside the tiny microscopic nucleus because it undergoes repeated coiling and supercoiling around the positively charged histone proteins. And DNA wrapped around the histone proteins forms a kind of a condensed, compacted structure inside the nucleus known as chromosomes. And we can be visualizing such chromosomes during the cell division, particularly during the metaphase stage of cell division. So here is a structure of a chromosome, and this chromosome is having 2 chromatids, which means this is a chromosome which has already completed with the S phase or the DNA replication phase, and because of which it has 2 of the chromatids. So in each chromosome, there is a region which is constricted, and this constricted region of the chromosome is referred to as centromere. And centromere is also the point where we have the 2 sister chromatids held together or joined together. So these 2 chromatids are referred to as sister chromatids because they are the exact copies, exact replica or duplicates of one another. So this chromatid is an exact copy of the second chromatid, or this chromatid is the exact copy of this chromatid. So they are known as sister chromatids. Now the region on either side of the centromere, so the region which is on either side of the centromere are referred to as chromosomes arms. So we have the region which is the shorter arm of the chromosome, and the shorter arm of the chromosome is called as P arm, whereas the longer arm of the chromosome is referred to as Q arm. So such is the understanding of a typical chromosome structure. Now in every normal human cell, we are going to be having 22 pairs of autosomes and 1 pair of sex chromosomes, because of which our 2n, our diploid chromosome number, becomes 46. Right? So we have a total of 23 pairs of chromosomes. And for a normal human, our genotype becomes 22AA plus XX if it is a normal female, or for a normal male, the genotype becomes 22AA plus XY, where AA is denoting the autosomal pair, and X and X denotes the 6 chromosome pair in females, or XY is the 6 chromosome pair in males. Look at this picture of the human chromosomes. This is referred to as human karyotype. The technique of pairing and ordering all the chromosomes of an organism is referred to as karyotyping. Now in this human karyotype, you can see that out of the 23 pairs of human chromosomes, the 22 pairs of autosomes look similar in both males and females, but the 6 chromosome pair, which is our last 23rd pair, differ between males and females. So since the males have one X and one Y as their 6 chromosome pair, so the X chromosome is relatively larger to the Y chromosome. And by looking at the human karyotype, you can predict with the sex of the individual. So we inherit one copy of each chromosome from our parents. Well, chromosomes are basically housing DNA or proteins. Let me just help you to recall some of the basics of DNA that you have learned last year. So DNA are chemically double-stranded polynucleotides. And each nucleotide, each monomer of the DNA is composed of 3 subunits, which are a deoxyribose sugar, a phosphate group, and one of the 4 nitrogen bases, which could be A, T, G, or C. Now let's look up here at the simple illustration of a DNA, double-stranded DNA, having only 2 nucleotides. Now you can see that each nucleotide is composed of a ribose sugar, a nitrogen base, and a phosphate group. The first carbon of the sugar is attaching to the nitrogen base, whereas the 5th carbon of the sugar attaches to the phosphate group. And the nucleotides in the DNA are held together by means of a bond which is known as phosphodiester bond. Now, the 2 strands of the DNA run in opposite direction, and they are said to be anti-parallel. Now, these 2 strands are held together or are stabilized in a DNA by means of hundreds of these hydrogen bonds between complementary base pairs. So, we learned about the base pair rule in DNA, where it states that if one of the strands of the DNA has adenine as its nucleotide, then the other strand, the opposite strand of the DNA, should be thymine. So adenine always pairs with thymine. Likewise, if one strand of the DNA has guanine as its nucleotide base, then the opposite strand should be a cytosine. So guanine always pairs with cytosine. So the base pair rule is A pairs with T, G pairs with C, and vice versa. And the number of hydrogen bonds between A and T will be 2, whereas the number of hydrogen bonds between G and C is 3. Now, the sequence of nucleotide or the sequence of base pairs in a DNA or in a gene, because gene basically is a segment of DNA. So the sequence of base pairs or the sequence of nucleotide in a DNA or in a gene would be serving as a blueprint for making all the proteins of our body. So basically, the sequence of the amino acids in a protein is dictated by the sequence of base pairs in the gene or the sequence of base pairs in a DNA. And hence, we say that DNA is the genetic material responsible for making all of the proteins of our body. Next, how do we have the expression of a DNA into a protein, or how do we explain the expression of the genes, which are actually a segment of the DNA, into a protein? So during gene expression, DNA is first copied or transcribed into an mRNA, and the mRNA is then translated into a protein. And this sequence of events describing the flow of genetic information is referred to as the central dogma of molecular biology, where this rule states that the flow of genetic information is DNA to RNA to protein. So what we need to understand here is that the gene or the DNA is not directly synthesizing protein. So DNA or the gene which are located inside the nucleus would be first copied into another molecule, and this molecule is referred to as messenger RNA. So it first makes a messenger RNA which carries all the information of the DNA or the gene, and then the messenger RNA would go out into the cytoplasm and then synthesize protein. And such kind of the genetic flow of information is the central dogma of molecular biology. And we are going to be learning about these events in detail in our next chapter. Now, to get a better understanding of this, let's take an example. We have a gene which is known as HBB gene. And this gene is located on our chromosome number 11. So it becomes an autosomal gene. And HBB gene is important because it is the gene responsible for making beta globin chain of hemoglobin. So we know we have a protein in our RBC known as hemoglobin, which is responsible for providing with the red coloration to RBC cells, as well as helping in the transportation of oxygen throughout our body. And a normal adult hemoglobin, which is referred to as HbA, is consisting of 4 polypeptides. So 2 of the polypeptides are alpha polypeptide chains, and the 2 other polypeptide chains are the beta polypeptide chains. So the beta polypeptides or the beta globin chains are produced by HBB gene. Now, a normal beta globin or polypeptide has 146 amino acids. But in this illustration here, we are just going to be looking into some of the first, I guess, 18 base pairs of the beta hemoglobin DNA or the gene. So, this is the normal sequence of HBB DNA, where the sequence reads as CAC, GTG, GAC, TGA, GGA, CTC. So these are the 18 base pairs of a normal HBB DNA, which then gets copied or transcribed into an mRNA. And when we have the copying of the DNA into an mRNA, the rule would be a complementary base polymerization, which is to say C would be complemented with, in the mRNA, it's going to be complemented with G. A would be complemented with U, because in RNA we don't have thymine. Thymine is replaced by uracil. Then C would be complemented with G. Likewise, T would be complemented with A. So we have a complementary base polymerization when we have the synthesis of mRNA from a DNA. So now, since this is the normal DNA sequence, the normal mRNA sequence copied from this DNA would be reading like this. So it has GUG, CAC, CUG, ACU, CCU, GAG. You need not be memorizing all of these sequences. They are just for your information. Now, from such a normal mRNA, we are to be synthesizing the protein, and the protein is beta globin protein. Now, what we also need to understand is that the sequence of mRNA is read in groups of 3 letters or 3 bases. So, each group of 3 bases forms a triplet, or it forms a codon of an mRNA. Okay, so in mRNA, we are going to be always reading the bases in groups of 3 letters or 3 bases, which are known as triplets or codon. Okay, so now here we have 1, 2, 3, 4, 5, 6 codons. These are the first 6 codons in an mRNA of a normal HBB gene. Now, such codons, as we know, or such the sequences are going to be determining with the sequence of amino acid in the protein. Because we said earlier, DNA, the sequence of base pairs in the DNA would decide the sequence of amino acids in the protein. So the sequence of the amino acid, so which 6 amino acids are going to be present or carried in a normal beta globin protein is determined by the sequence of mRNA or the codons in the mRNA. And each codon would be coding for a single amino acid. Okay, so now GUG is the first codon or the first triplet which codes for valine. Likewise, CAC codes for HIS, CUG is coding for LEU, ACU is coding for THR, CCU is coding for PRO, and GAG is coding for GLU. Okay, and these are the first 6 amino acid sequence of a normal beta globin gene. So in this way, what we see here is that there is the genetic flow of information from a DNA to mRNA to protein, and the sequence of the DNA or the sequence of the mRNA or the codon in the mRNA would be ultimately determining with the sequence of amino acid in the protein. In this way, the human genome is estimated to be having a total of 20,000 to 25,000 genes. And DNA, being a highly stable genetic material, is able to transmit these genetic information from one generation to the next generation without any change most of the time. However, some changes can occur occasionally, and such changes in the genetic material is referred to as mutation. Let me now come back to this example of HBB gene in humans, where we know that HBB is responsible for producing beta chain in hemoglobin. A normal HBB gene without any mutation would result in the formation of a normal beta chain or a normal hemoglobin. And a normal hemoglobin is represented with HbA. So HbA becomes the normal allele for hemoglobin. Now, in a normal HBB gene, the DNA sequence reads as CTC. So you can ignore these sequences, but you have to know the sequences which I have written in red color. So the normal DNA sequence in the HBB is CTC, which would be then copied into an mRNA, and the mRNA codon of CTC reads as GAG. And this is the 6th codon. So please keep in mind the position of the codon. So this is the 6th codon, which is in a normal mRNA of HBB gene. And GAG codon would then be coding for a particular amino acid, and the 6th amino acid coded by GAG codon is GLU, which stands for glutamic acid. So a normal GAG codon is coding for glutamic acid at the 6th position. And this results in the formation of a normal hemoglobin, which is denoted as HbA. Now let's look into another situation here where there is a mutation that has happened in HBB gene. And this mutation is denoted as HBS. So HBS actually means it is a sickle-shaped hemoglobin. Okay, so the mutation has resulted into the change in the structure of RBC. Instead of the normal disc-shaped structure, rounded structure, now RBC has changed into a sickled shaped or a crescent shaped structure, and such hemoglobin is denoted as HBS. So now let's look into what went wrong in these HBS individual. Now, instead of the sequence CTC in their DNA, the HBB mutated DNA has CAC. So there's a change in just a single base. Okay, CTC has become CAC, and CAC is then copied as GUG instead of GAG. So the sixth codon in the normal mRNA was GAG, but the sixth codon in the affected or mutated mRNA of an HBS mRNA is GUG. So we have the substitution of A in the sixth codon by U. And this single base substitution has resulted in a change in the sixth amino acid as well. So we understand that the sequence of the DNA or the sequence of the mRNA would be determining with the sequence of amino acid in the protein. So now in the HBS beta globin chain, instead of glutamic acid at the sixth position, now we have glutamic acid replaced by valine, another amino acid, because there was a change in the codon. So the codon has now become GUG, and because of which the amino acid which is at the sixth position is not glutamic acid, but it has now changed to valine. And this kind of mutation has resulted in this kind of the substitution of glutamic acid by valine because of a single base substitution in the GAG codon into GUG codon in the HBS mRNA. It has resulted in the complete, you can say, change in the structure of hemoglobin. So now the hemoglobin or the RBC has become sickled shape, and such kind of hemoglobins are represented as HBS. And an individual having two of such alleles, two copies of HBS in their gene is going to be having a disease which is known as sickle cell anemia. Over a lifetime, DNA can undergo mutation. Mutation refers to change in the DNA sequence which results in changes in the genotype and phenotype of an organism. Now, there's a misconception that mutation can only occur in few individual or organisms and that mutations are always bad. We need to think again because mutation can occur in everyone. We have to know that there are hundreds of mutations that occurs during DNA replication. Fortunately, these mutations are corrected on its own through a very efficient DNA repair mechanism. And not all mutations are bad or harmful. Mutations can be beneficial because it can be increasing good genetic variation within species or in a population. And mutation is indeed a driving force for evolution. So, certain percent of mutation or frequency of mutation can occur spontaneously in an organism, such as during DNA replication. But most of the mutations are induced mutation, which refers to mutations which are caused by some external agents. And these agents which are causing mutations are collectively referred to as mutagens, which could be physical, chemical, or biological mutagens. So physical mutagens includes all of the harmful ionizing and non-ionizing radiations such as X-rays, beta rays, gamma rays, or UV rays. And chemical mutagens are all the harmful carcinogenic chemical compounds such as formaldehyde, nitrosamines. Nitrosamine is a carcinogen present in tobacco. And biological mutagens include some of the organisms which can be causing infections like viruses. So we have HPV virus, which we know, which can be causing the cervical cancer in human females, as well as we have bacteria which can be causing mutations or cancer. So mutations which are harmful can result in many different types of diseases, which are collectively referred as genetic disorders. Let me now discuss the genetic diseases which are caused due to mutation in the gene or chromosome of an individual. And these diseases are collectively referred to as genetic disorders. Now, there are three broad categories of genetic disorders. The first type is known as monogenic disorder. This refers to a disorder or a disease caused due to mutation in only one gene, monogenic. It's only the mutation in a single gene, and because of which such disorders are also called as Mendelian disorders. The second type are known as polygenic disorders. Now, in this type of disease, there is a mutation in multiple genes, or there is mutation in more than one gene. That becomes polygenic disorders. And the third type are known as chromosomal disorders, where it refers to all the aberration or abnormalities in terms of chromosome number, or some kind of aberration in terms of chromosome structure. So collectively they are known as chromosomal disorders. Now, in this chapter, we are to be focusing on Mendelian disorders or monogenic disorders and chromosomal disorders. Monogenic disorders and polygenic disorders are caused due to gene mutations. And gene mutations refers to any kind of a mutation in the sequence of the gene or the sequence of DNA. Now, gene mutations can be caused due to substitution, deletion, or insertion type of mutations. Now, let's look into these three types of mutations. Substitution mutation, as the name implies, we have a substitution or a replacement, and such substitution can be a substitution of one or more of the nucleotides or bases in the DNA or in the RNA of the gene. Deletion mutations refers to a mutation where one or more nucleotides or one or more bases in the DNA or in the gene or in the RNA are removed or are deleted from the sequence. And insertion mutation refers to addition of one or more nucleotides or one or more bases into the DNA sequence or into the RNA sequence of the gene. And all these three types of mutations can be resulting in gene mutation. Now, let's study this in a simple example here. So this is just a hypothetical example. Here I have shown a normal DNA sequence which reads as TAC, CTC, CTC, CAC. So this normal DNA is synthesizing an mRNA which reads as AUG, GAG, GAG, GUG. So mRNA are always to be read in groups of 3 bases or triplets, right? And here you can be seeing 1, 2, 3, 4 of the triplets. So this mRNA is having 4 triplets or 4 codons. And such 4 triplets or codons are going to be coding for 4 amino acids. And the amino acids are of the protein are MET, GLU, GLU, and VAL. So let's assume that such is a hypothetical normal DNA or a gene producing a normal protein. Now, let's try to study how the effect of substitution, deletion, and insertion can be seen in this kind of a gene or a DNA. Now, what I've done is, in this normal DNA sequence, to study the effect of substitution, I have substituted only one base, and that base is this T. So this T has been substituted by an A, and that substitution has now changed the sequence from this to CAC, CAC. Okay, so and then this is going to be resulting in the synthesis of an mRNA having also a change in its third codon. So here the third codon, instead of GAG, has now become GUG, right? And this substitution in the DNA has resulted also in the effect in the amino acid sequence. So instead of GLU, here in the third codon GUG is now coding for VAL. So now the sequence of amino acid has become MET, GLU, VAL, VAL. So what you see is there's a change in one of the amino acid sequence of this kind of the mutation, which is a single base substitution mutation. And this is therefore not the normal protein because it's not having this type of the amino acid sequence. It's having a different type of amino acid sequence. So such kind of proteins are going to be a mutant protein. Likewise, let me give you an example of one base deletion. So in this case, what I've done is I have deleted this. Okay, so now if we have a deletion of the fourth nucleotide, then the DNA sequence would read as TAC, TCC, TCC, AC. And because of which the mRNA sequence are going to be AUG, AGG, AGG, UG. So because of the deletion of a single base, from the point of deletion, from this point onward, now there is the change in the codon. Now codons, the second codon was GAG in the normal mRNA, but now it is AGG. Third codon was GAG again, here it is AGG. So in this way, we see that the second and the third amino acid in such a one base deleted protein is again different from the normal protein. And because of which this is yet another type of a mutant protein resulted due to the single base deletion. Likewise, if we have adhesion, okay, now I have taken the example of a single base adhesion. So if we have an addition of T, now this is a new addition into the DNA sequence. So T has been added in between these two C. T has been added into this sequence between the two C's here, and that has resulted in the sequence TAC, TC, 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 CAC. And the mRNA is reading as AUG, GAG, AGA, GGU, and only C. So what you can see again is from the point of insertion, from the point of this addition, there is a change in the codon sequence. Now the third codon, instead of GAG, it is AGA. Fourth codon, instead of GUG, it has become GGU. And due to which we can be finding two different kinds of amino acids being coded by these two different codons. So the amino acids are MET, GLU, ARG, and GLY. And that results in an abnormal type of protein or a mutant protein. So such as an example of one base substitution, one base deletion, and one base addition in this normal DNA sequence, resulting in mutant protein or abnormal protein. And these kinds of mutations, which are because of just a single change in the base or in the nucleotide of the DNA, which could be a single base substitution or a single base deletion or a single base addition, are collectively known as point mutation. So mutations in which only one base or one nucleotide in the DNA sequence gets affected. Either it is substituted, or it is deleted, or it is added or inserted into the DNA. Such kind of mutations are collectively referred to as point mutation. Now, all of these are examples of point mutation. Point mutation because of one base substitution, point mutation because of one base deletion, point mutation because of one base insertion. But if we look into the effects of these different types of point mutation, we see that the effect is more serious or more greater felt in cases of deletion and insertion. So with the deletion of one base, it has resulted in a complete change in the codon from the point of deletion of that particular nucleotide or base. Likewise, the addition of a single base or insertion of a single base, from the point of insertion, we can see that the entire reading frame, the entire codon sequence got affected. Okay, and such kind of mutation which causes some kind of a change in the reading frame of the mRNA or reading frame of the base sequence is referred to as frameshift mutation. So deletion and insertions are two examples of frameshift mutation, because from the point of deletion, or from the point of insertion, we find that the reading frame of the base sequence or the reading frame of the mRNA codon gets affected. Let me now discuss Mendelian disorders or monogenic disorders by taking specific examples of human diseases. These are the diseases which are caused by mutation in one gene. And these diseases follow a similar pattern of Mendelian inheritance. Either these diseases are completely dominant or they are completely recessive. Here are an example of autosomal recessive Mendelian type of disorders, which are sickle-cell anemia and thalassemia. Now let me first take up sickle-cell anemia. As we have seen earlier, sickle-cell anemia is caused by a point mutation. So the sickled hemoglobin is caused due to the substitution of glutamic acid by valine in the 6th amino acid position of beta globin protein. And this kind of the substitution of glutamic acid by valine happened because of the substitution of one base at the 6th codon of the beta globin mRNA. So you can describe sickle cell anemia in terms of substitution of one amino acid or in terms of substitution of one base in the 6th codon of the mRNA. Now, such kind of a single substitution has resulted in a complete change in the phenotype or the structure of hemoglobin. So, these individuals are going to be producing sickled hemoglobin or sickled RBC. And the allele for sickled hemoglobin is denoted as HbS, and the normal allele, which is producing normal RBCs, is represented as HbA. Now, the reason why sickle cell anemia is considered as autosomal disease is because the gene for hemoglobin, the HbB gene, is located on chromosome 11. And we know that human chromosomes from 1 to 22, the first 22 pairs of human chromosomes are all autosomal chromosomes. And because the gene is located on the autosomal chromosome, it becomes an autosomal linked disease. And if we look into the pattern of inheritance, now since we know that each individual is going to be carrying 2 copies of a gene, and in a human population, we can be expecting 3 different types of genotypes for hemoglobin. So individuals can be having homozygous normal hemoglobin type of the genotype, which is HbA, HbA. So if individuals have homozygous HbA, HbA type of genotype, their phenotypes are all going to be normal. That means they are going to be producing normal shaped RBCs or hemoglobin. Individuals with heterozygous genotype. Now these individuals are going to be having 2 dissimilar alleles for the same gene. So they have normal HbA allele as well as the sickled HbS allele. And because of which these heterozygous individuals, even though they have the HbS allele, their phenotype is a normal phenotype. That means they are still capable of producing normal shaped RBCs or hemoglobin. And this itself is an inference that HbS allele is recessive to HbA. In the presence of HbA, HbS is unable to express itself. To develop a sickle cell anemia in an individual, such individuals are to be having 2 copies of HbS. So when HbS exist in homozygous combination, then individuals are going to be suffering from sickle cell anemia. So their RBC, the phenotype would be changed, and they have sickled RBCs or sickled hemoglobin. And hence the disease is referred to as autosomal and recessive type of Mendelian disease. Individuals with such sickled RBCs are going to be showing many complications, of which, unlike the normal RBCs, sickled RBCs tend to be more rigid and stickier. They make lots of clumps in the blood, and they stick to the walls of the blood vessels, thereby resulting in the reduced supply of oxygen to all of our major organs. And that's the reason why one of the major symptoms of this disease is anemia. Now, yet another example of autosomal recessive disease is thalassemia. As I said earlier, a normal adult hemoglobin has 4 polypeptides, 2 alpha polypeptide chains and 2 beta polypeptide chains. And we know that the beta polypeptide chains are synthesized by a gene located on chromosome 11. Likewise, the two alpha polypeptide chains are synthesized by two genes which are located on chromosome number 16. And the names of these two genes are HBA1 and HBA2 gene. Now, unlike sickle cell anemia, in thalassemia, individuals might be showing mutation in either the HBB gene, which is located on chromosome 11, or they might be showing a mutation in the HBA genes, which are located on chromosome 16. And depending on which gene is affected, we have two types of thalassemia: alpha thalassemia and beta thalassemia. So alpha thalassemia, alpha in reference to the alpha chain. And we know that alpha chain is the HBA gene. So a disease, alpha thalassemia, is a disease caused due to the mutation in HBA genes resulting in alpha chain deficiency in hemoglobin. So because of a mutation in these genes, HBA genes, which are located on chromosome number 16, these individuals are going to be making very low amounts of alpha chain, and this would be consequently resulting in very low levels of hemoglobin and very low levels of RBC in the individual. And such type of a disease is called as alpha thalassemia, because the gene affected is the gene which is responsible for making alpha chains. The second type of thalassemia are known as beta thalassemia. In individuals with beta thalassemia, here this is caused by mutation in HBB gene, which is located on chromosome 11, and thereby it results in beta chain deficiency. So these individuals are producing lower amounts of beta chain, and they might be having higher production of alpha chain, and that would be indirectly resulting in the reduced level of hemoglobin or the reduced level of RBCs. And this kind of the disease is referred to as beta thalassemia. So both these types of alpha and beta thalassemia are resulting in a reduced level of hemoglobin. We have an imbalanced level of the hemoglobin chains being produced in alpha and beta thalassemia. Alpha thalassemia are going to be having higher of beta chain and very less of alpha chains, and hence they are going to be having the abnormal levels of hemoglobin. Likewise, in beta thalassemia, they are going to be showing very high levels of alpha chains, but then very low levels of beta chain, thereby again resulting in reduced level of a normal hemoglobin. And since these genes which are making the alpha and beta chains are present on our autosomal chromosome, so thalassemia is yet another example for autosomal linked disease. And if we look into the inheritance of thalassemia, individuals who have two copies of the mutant HBA gene or the mutant HBB gene will only be expressing with thalassemia condition. So individuals with a heterozygous genotype, right, having one of the normal HBA allele and the other the defective allele, in a heterozygous combination, these diseases cannot be making their expression. So heterozygous individuals show normal phenotype. That means they're going to be making normal amounts of alpha and beta genes. And because of which thalassemia, in order to have the expression of the thalassemia, the individuals are always to be having homozygous recessive combination, genotypic combination, and due to which thalassemia is an autosomal recessive disease. Another type of autosomal disorders are the autosomal dominant diseases. So one such example that I've taken here is Huntington's disease. Huntington's disease is a progressive brain disorder which is caused by mutations in HTT gene. And this gene HTT is located on our 4th chromosome. Now HTT gene is, mutation in the HTT gene is responsible for excessive production or excessive accumulation of Huntington protein in our brain. And excessive accumulation of Huntington proteins causes breakdown of nerve cells, and thereby it results in multitudes of symptoms or multitudes of effects on the individual who suffer from Huntington's disease. So the major symptoms associated with Huntington's disease are uncontrolled movements, thinking impairments, and emotional disorders. Now, because HTT gene is located on our autosomal chromosome, this is an autosomal linked disease. And if we look into the pattern of inheritance of the disease, individuals can be expected to be having 2 copies of normal allele without any kind of mutation in the HTT gene. Or individuals can be expected to be having 1 copy of the normal allele for HTT and 1 copy of an affected or a mutated HTT allele. Or individuals can be having 2 copies of the mutated HTT allele. So mutated allele is represented with capital H and normal allele is represented with small h. So here are the 3 possible genotypes. First one is homozygous capital H, capital H. So these individuals have 2 copies of Huntington's allele. And because they have 2 copies of Huntington's allele, individuals have the expression of the phenotype or the disease. So they suffer from Huntington's disease. Now those individuals who are heterozygous, which means these individuals are having 1 copy of Huntington's allele and 1 normal copy. So individuals who have heterozygous genotype also are expressing with the disease phenotype. So they are also suffering from Huntington's disease. So this gives us the information that Huntington's allele is dominant over normal allele. So our normal allele in the presence of Huntington's allele cannot be making its expression, and because of which Huntington is a dominant disease. And individuals who have 2 copies of normal alleles, which are small h and small h. Now because normal alleles are recessive and because they are existing in homozygous combination, such individuals are all going to be normal individuals producing normal levels of Huntington protein. So this is a disease which talks about how we have the autosomal dominant pattern of inheritance in humans. Another category of Mendelian disorders are the sex-linked disorders or sex-linked diseases. And these are the diseases which affects differently in males and females. And the genes for these diseases are located on sex chromosomes. Now I'm going to be focusing on 2 of the X-linked recessive diseases. And you will notice that most of the diseases, sex-linked diseases, are recessive and they are X-linked diseases. Now we do have some Y-linked disease or sex-linked dominant diseases, but then they occur in lower frequency or they are much rarer in human population. So Y-linked diseases or sex-linked dominant diseases are considered to be more lethal, and embryos showing such mutations cannot be surviving to full term. It undergoes spontaneous abortion. Now the 2 of X-linked recessive diseases that we will learn are hemophilia and red-green color blindness. Hemophilia is a bleeding disorder which is caused by mutations in F8 gene, which is located on the X chromosome. And because of the mutation in the F8 gene, these individuals produce very low amounts of clotting factor 8. So F8 gene is responsible for synthesizing clotting factor 8, which is very much essential for blood clotting. Now, because individuals with hemophilia have very low amounts of clotting factor 8, they are not going to be having proper clotting whenever there is a cut or an injury to the blood vessels. So this would be resulting in prolonged bleeding or excessive bleeding. Since the gene is located on the X chromosome, so it is said to be an X-linked disease. And when we look into the pattern of inheritance of hemophilia, we see that it is a recessive disease. So to understand this, let's look into the possible genotypes and the phenotypes of the individual. Now, whenever we have to write the genotypes of sex-linked diseases, we have to show also the sex chromosomes carrying the alleles, because sex chromosomes are different in males and females. Females have 2 X chromosomes, whereas males only have 1 X chromosome, and the other sex chromosome is Y chromosome. So females can be having 3 possible genotypes. The genotypes are where both of the X chromosomes are carrying the normal allele, and normal allele is denoted with capital H. So both of her X have the normal capital H allele. So she becomes homozygous for normal allele, and her phenotype is also normal. Right? She is showing normal blood clotting. Females who have heterozygous genotype, where one of her X chromosomes carry the normal allele, capital H, whereas the other X chromosome carries the hemophilia allele. So hemophilia allele here is denoted with small h. And when the females are heterozygous with capital H and small h, phenotypically she is still normal. She is able to show normal blood clotting. So this itself is telling us that hemophilia allele in the presence of the normal allele cannot be making its expression. So hemophilia is a recessive disease. Whereas females who have 2 copies of hemophilia allele or 2 copies of the recessive small h allele, then such females, homozygous small h, is going to be suffering from hemophilia. So hemophilia can only be affecting to a female when she carries 2 of the recessive alleles for hemophilia. So when both of her X chromosomes has the recessive hemophilic allele, then only female would be suffering from hemophilia. When we look into the males, males only have a single X chromosome, and the other sex chromosome is the Y chromosome, which cannot be compensating for the X chromosome. And because of which, males who have the normal allele on the only X chromosome that he has received from his mother would always be having normal blood clotting. Whereas males who have received an X chromosome from his mother carrying the recessive hemophilia allele would be suffering from hemophilia. So only a single copy of the hemophilia allele carried on its own on his only X chromosome would be making this male hemophiliac, whereas a single copy of the hemophilia allele in the females will not be making the females hemophiliac, because females have 2 copies of X chromosomes. And because of which females are at an advantage here in terms of sex-linked diseases, especially the X-linked diseases, because females have 2 X chromosomes. So if one of the X chromosomes have the defect, but the other X chromosome have the normal allele, then she will be having the compensation by that other X chromosome and will not be suffering from the disease. Whereas most of the X-linked diseases are more commonly or more, there's a higher risk of the males having the expression of X-linked diseases, because they only carry a single X chromosome. Another example for X-linked recessive disease is red-green color blindness. Now, this is a condition where individuals have problems in differentiating between the shades of red and green color. Now, we know that our retina has 3 of the principal cone cells, red, green and blue cone cells. Now, in individuals with red-green color blindness, they have a mutation in 2 of the genes responsible for red cone and green cone. The gene which is responsible for red cone is called as LW gene. Actually, it is OPN1 LW gene, and the gene responsible for green cone is MW gene or OPN1 MW gene. So longer wavelength, LW is longer wavelength, MW is the medium wavelength gene, and both of these genes are located on X chromosomes. Now, when there is a mutation in the LW and MW genes in red-green colorblind individuals, they are going to be having very low amounts of deficient amounts of red cones and green cones, and because of which they have a problem differentiating between green and red shades. Now, here's an image where you can see in the background of the red coloration, there's a number actually given in green color, as well as in the background of a green coloration, you have a number which is given in red color. So most of us can be easily identifying the number or seeing the number, but people who have red-green color blindness might be having a problem in differentiating the or identifying the number in this particular background. So now when we look into the nature of the inheritance or the pattern of inheritance of red-green color blindness, what we see is females having 2 copies of the normal color gene, which is capital C, capital C. So capital C is denoting with the normal allele. So females who has capital C on both of her X chromosomes are showing normal color vision. Whereas females who are heterozygous, which means one of her X chromosomes is having the capital C normal allele and the other X chromosome is having the small c, that is red-green colorblind allele. And a female with heterozygous capital C small c genotype is still normal in her color vision. Alright, so this itself is indicative that colorblind allele is recessive to normal allele. So we are denoting colorblind with small c letter and the normal allele with capital C letter. Now, a female can only be affected with color blindness when both of her X chromosomes have small c, small c. So then only she will be having color blindness. But when we look into the males again, since they have only one X chromosomes, so the X chromosome having capital C allele would be resulting into a normal color vision male, whereas males having small c on his only X chromosome is going to be affected with colorblind. So in this way, what we get to see here is X-linked recessive diseases or the X-linked diseases in general are more commonly affecting the male populations than the female population. And the reason is because males have only one X chromosome, whereas females, because we have 2 copies of the X chromosome, so even if we have a single copy of the mutated or the defective allele, we can still be compensated by our other X chromosome, which can carry the normal allele. And hence, in the heterozygous state, females are not affected with X-linked diseases, whereas males who only have one X chromosome with only one copy of the defective or the recessive allele are going to be also expressing with the disease. Unlike Mendelian disorders, some of the genetic disorders are caused by chromosomal mutations, which are caused by the abnormalities or aberrations in chromosome number or in the chromosome structure. Now, structural abnormalities in the chromosome are caused by deletion, duplication, inversion, insertion, or translocation types of mutations. Now, to get a brief idea about these mutations, let's check into the figure here. So, in deletion mutation, a portion of the chromosome is deleted or missing. So, you see that the chromosome has become a little shorter. In duplication mutation, a portion of the chromosome is duplicated. Hence, the chromosome appears a little longer. In inversion mutation, a portion of the chromosome has broken off, turned upside down and reattached itself to the same chromosome. So actually it is a particular portion where the chromosome got inverted. And in insertion mutation, a portion of the chromosome is deleted from one chromosome and is then inserted in between another chromosome. In translocation, a portion of one chromosome is transferred to another chromosome. So these are some of the mutations which can be resulting in structural aberrations or structural changes in the chromosome. Abnormal chromosome number can be of 2 major types, Aneuploidy and Polyploidy. Aneuploidy refers to a condition where a part of the chromosome set is changed. So since we have 2 copies of each of the 23 chromosomes, so in aneuploids, one of the chromosomes might be having either more than 2 copies of it, or it can be having less than 2 copies of it. And such conditions are referred to as aneuploid. Whereas polyploidy refers to a condition where there's a change in the whole set of chromosomes. So the whole set of the chromosome, that is from chromosome 1 to chromosome 23, they are going to be having in extra copies. So an individual might be having, let us say, 3 copies of each of the 23 chromosomes with a total of 69 chromosomes. Or they can be having 4 copies of each of the 23 chromosomes with a total of 92 chromosomes. Now, such kind of Polyploid humans are unviable. It is a kind of a lethal mutation. But polyploidy is very common in flowering plants. So now what causes aneuploidy and polyploidy? The main reason for aneuploidy and polyploidy is because of a phenomenon known as non-disjunction. So during the time of, we know that during the time of cell division, there is segregation of the chromosomes, right? So chromosomes are to be equally segregating into daughter cells. So during the time of mitosis or during the time of meiosis, when we have the gamete formation taking place. So there might be some failure in the segregation of chromosomes. So failure in the segregation of homologous chromosomes during meiosis 1, or failure of the segregation of the sister chromatids during meiosis 2 or during mitosis can be resulting in non-disjunction, right? And in aneuploidy, it is the non-disjunction which can be occurring in certain chromosomes, in some of the chromosomes, not all of the chromosomes, whereas polyploidy is a total non-disjunction. Finally, 3 examples of aneuploidy in humans are Down syndrome, Klinefelter syndrome, and Turner's syndrome. Both Down syndrome and Klinefelter syndrome are caused due to a gain in the chromosome. So both of these have 47 chromosomes. Whereas Turner syndrome is caused by a loss in the chromosome, and because of which their total chromosome number is 45. The genotype of a Down syndrome patient can be written as 44 plus 1 plus XX or XY. So Down syndrome can be affecting both females and males equally. And the extra copy of the chromosome that they have in their autosome is the 21st chromosome. So in Down syndrome patient, they have 3 copies of the 21st chromosome, unlike the normal 2 copies. And because they have 3 copies of 21st chromosome, Down syndrome is also called as trisomy of 21. Now, how such kind of 3 sets or 3 copies of 21st chromosome came into the individual? Now, this occurred because of non-disjunction. And non-disjunction, as I stated earlier, is an event where we have the failure of segregation of the pair of chromosomes. So the pair of 21st chromosome fails to segregate during the time of gamete formation, and this could be happening during the egg formation, or it could also occur during the time of sperm formation. Now, because the 21st pair could not be segregating or separating equally into the eggs or into the sperm gametes, so some of the gametes are going to be carrying both the copies of the 21st chromosome. And what I've shown here is, I've shown here an egg having 2 copies of 21st chromosome. So this egg is having rest of all other chromosomes in only 1 copy in the haploid set, whereas the 21st chromosome, because of the failure of segregation, because of non-disjunction, so this is a non-disjunction egg where the egg is carrying both the copies or 2 copies of the 21st chromosome, and such an egg has fused with a normal sperm, and because there is also 1 copy of the 21st chromosome in the normal sperm, so this results in, such kind of a fertilization results in 3 copies of 21st chromosome, and that is what we call as trisomy of 21, or the disease is or the condition is considered as Down's syndrome. In Klinefelter syndrome, they, as we know, they have an extra of an X chromosome. So their genotype is 44 plus XXY. So this extra copy of the X chromosome has resulted in the trisomy of sex chromosome. So they have 3 sex chromosomes, where the extra sex chromosome is the X chromosome here. And these individuals are going to be, Klinefelters are always going to be affected in the males. Okay, so it's a kind of a genetic condition disorder which is of the males, where they have an extra copy of X chromosome. And again, why such kind of an extra X chromosome has resulted in the Klinefelter's individual? This is because of non-disjunction of the sex chromosomes. So during the time of egg formation or during the time of the sperm formation, what has happened is two of the sex chromosomes, the pair of sex chromosome, fail to segregate equally into the gametes, daughter cells, and as a result of which we can be having X with both of the copies of the sex chromosomes, or we can be also having sperms having both of the sex chromosomes. So you have here an egg which has two of both of the X chromosomes. If such a kind of an egg, abnormal egg having two of the X chromosomes fuse with a Y chromosome containing, let us say, sperm, then this is going to be resulting in XXY condition. Or in another way, we can be having sperms having both of the sex chromosomes, X and Y. And if such an abnormal sperm fuses with a normal egg, then again this is going to be resulting in the combination XXY, which is the condition called as Klinefelter's syndrome. And here we have trisomy of a sex chromosome, where the extra chromosome is the X chromosome. And these kind of the genetic conditions are always seen in males. The third example is Turner's syndrome, where there is a loss of one chromosome. And their genotype can be written as 44 plus X0. So they have a loss of one of their X chromosomes. And because of which Turner syndrome is always going to be affecting the females. So because of one less of the X chromosomes, the total chromosome number becomes 45. And this condition is also called as monosomy of X chromosomes or monosomy of sex chromosomes. So they only have monosomy in the sense they only have one copy of X chromosome. Now, this kind of monosomy of the X chromosome can be resulting in an individual because of non-disjunction during the gamete formation. So, as we know, the failure of the segregation of the sex chromosomes during the gamete formation can result in formation of egg with two copies of the sex chromosomes and another type of egg with no copies of sex chromosome or no sex chromosome at all. So if such kind of an egg having no sex chromosome has fused with a normal X chromosome containing sperm, then again it would be resulting in the genotype X0. Or if we have the fusion of an abnormal sperm having no sex chromosome because the sex chromosome failed to segregate equally. So both of the X and Y went into one gamete, and this sperm failed to receive the copy of sex chromosome. And when such kind of sperm having no sex chromosome fuses with a normal egg, again the genotype is going to be becoming X0. And these are going to be resulting in the condition which is called as monosomy of X chromosomes, or it is the condition which is called as Turner's syndrome. These three aneuploidy conditions in humans are usually not an inherited disease because it's not caused by any kind of a mutation in the gene. Rather, it is caused by the random non-disjunction of chromosomes during gamete formation. And such kind of genetic conditions can be resulting in multitudes of symptoms in the affected individuals. Now, to list some of the major symptoms of these three conditions, let's look up at the figure here. So individuals affected with Down syndrome are going to be showing more flattened face and nose feature, and they have short and broad hands with very short fingers. And their first and second toes are widely separated, and they have a characteristic protruding tongue and a partially opened mouth. And they also show a kind of a characteristic palm crease. Such individuals affected with Down syndrome might also develop congenital heart diseases, and they also show retardation in physical and mental growth. Individuals affected with Klinefelter syndrome have a taller than average height, and they have very long arms and legs, and very less hair on their face and body, unlike the normal males. And their testicles, or their testes and penis, are highly underdeveloped, and because of which they become sterile males. And they also show some kind of feminine characteristics, like we can be seeing enlargement of breast in these males, which is a condition called as gynecomastia. And they also have increased belly fat. Now, females affected with Turner syndrome are normally short-statured, and they have a very highly underdeveloped ovaries. So the ovaries are rudimentary, and because of which such females are again sterile females. And they also have underdeveloped breasts with widely spaced nipple. And another very characterizing feature of Turner syndrome is that they have a webbed neck. With this, I come to the end of today's video lesson on mutation and genetic disorders, and I'll catch up with my next video lesson on pedigree analysis.